Hi, my name is Alexa Renna, and the energy system I chose to do my presentation on is the ATP PCr system. I will be discussing how it works and training benefits about this system throughout my presentation. The basic energy systems. So before I discuss the main components of the ATP PCr system, I wanted to discuss all three systems. So cells can store only limited amounts of ATP, and they must generate new ATP consistently to provide the needed energy for cellular metabolism. So cells will generate ATP through one or more of these three metabolic pathways, which is the ATP PCr system, the glycolytic system, and the oxidative system. So the first two systems work in the absence of oxygen, also known as the anaerobic system, and the third system does require oxygen, which is also known as the aerobic system, which means requires oxygen. So I posted this little picture because it explains the different exercises and how those energy systems work for those, which I'll be discussing more throughout my presentation as well. So as you can see, ATP PCr leans more as lifting a weight, running, basketball. Later on it goes more into the glycolytic system. And then, as you can see, skiing or something that's more for endurance activity leans more towards the oxidative or aerobic system. What is the ATP PCr system? How does it work? So basically, the ATP PCr system is one of the simplest energy systems. It stores a very small amount of adenosine triphosphate, also known as ATP. Sorry if I mispronounce anything. And cells contain another high-energy phosphate molecule which stores energy, which is phosphocreatine. The simple pathway involves a donation of a PI from PCr to ADP to form ATP. Energy released from the breakdown of PCr is not directly used for cellular work. However, it is instead regenerating the ATP to maintain a constant supply, and this constant supply is even beneficial during resting conditions. The release of energy from PCr is then catalyzed by the enzyme creatine kinase, and then the energy released from the ATP splits a phosphate group, which prevents ATP depletion by breaking down PCr. This breaking down of PCr provides energy and PI to perform ATP from ADP, which provides energy. If you follow the principles of negative feedback and rate-limiting enzymes, creatine kinase activity is enhanced when concentration of ADP or PI increases. However, it is inhibited when ATP concentration increases. So when intense exercise has begun, the smallest amount of ATP in your muscle cells is broken down for immediate energy for the body, which will yield ADP and PI. This increased amount of ADP concentration will increase the creatine kinase activity, and the PCr is then catabolized to form more ATP or more energy for your body. Unfortunately, as exercise progresses and ATP is generated by two other energy systems, the creatine kinase activity is then inhibited. As you can see, this is a diagram of how the ATP PCr system works. So ATP is required for the man to lift the heavy weight or to do the squat in the picture. So because of this, we're getting a donation of PI, PCr, and ATP to form more ATP for the man to lift or squat a heavy weight. How it works. So the ATP PCr system has a limited capacity to generate ATP for energy, which will only last a few seconds. So the ATP PCr system can provide energy at a fast rate, but has a very low capacity for energy production, which thus supports exercise that is intense but of very short duration. For example, during the first few seconds of intense muscular activity, this could be sprinting, ATP is maintained at a relatively constant level. However, PCr declines steadily and is then used to replenish depleted ATP. When the body hits exhaustion, both ATP and the PCr levels are low and unable to provide energy for further muscle contraction and relaxation. So in this case, the capacity to maintain ATP levels with energy from PCr is limited. The ATP PCR system has been found to decrease within 1.3 seconds after the start of a contraction. Many researchers actually believe that the majority of the ATP PCR system is utilized and lasts about 10 to 15 seconds. Others say the duration of ATP PCR is 3 to 15 seconds. So I guess it may depend on the intensity of the exercise, but it is really interesting to learn about. As you can see in this graph, you can see the ATP and the PCR exhaustion. It hits at this case, 14 seconds, which is basically the duration of how long the system will work for that intense muscular activity. So I thought this chart was very useful in explaining that and also showcasing that. The ATP PCR system is predominantly used to resynthesize ATP during high-intensity and short-duration exercise. So the primary emphasis for the short sprints is on training the ATP PCR system. Training the system requires training at very high percentages of the athlete's HR max. The same goes for the anaerobic glycolytic system. Activities that emphasize maximum muscle force production, like sprinting and weightlifting events, rely most heavily on the ATP PCR system for energy. Other exercises include doing a push-up, lifting a weight, throwing a hammer, or swinging a golf club. Some sports that involve the ATP PCR system is golf, baseball, basketball, diving, track, and many others as well. I did post this little chart in this slide to showcase the other sports that involve more of an anaerobic approach than aerobic. So as you can see, like basketball, tennis, volleyball, fencing, field events, those lean more towards the anaerobic side. So more toward the ATP PCR and the glycolytic system. Soccer and hockey are more in the middle, but then you can see like skiing, swimming, 1.5K, rowing, and distance running, not sprints, is more towards the aerobic, the endurance side of things. I also added this image because I thought it was really interesting. It states sports like basketball, large amounts of energy are needed from aerobic and anaerobic sources and muscles. Anaerobic energy from glycolysis and the phosphocreatine allow for quick and powerful movements like bursting, jumping, and sprints. I really thought that those two components were really interesting in explaining the ATP PCR system and even the other anaerobic system. And for the aerobic system, it says that it helps during periods of recovery, like light running, jogging, stuff like that, more for endurance. So you can see the difference of all the energy systems in this one picture. Alterations in energy system contribution following upper body sprint interval training. So I did choose one of the studies to discuss in my presentation. So this study determined the results of sprint interval training, or SIT, which is a form of high-intensity training. SIT has shown to result in enhanced glycolytic enzyme activity and maximum anaerobic power. So it has been determined that 10 seconds of work utilizes aerobic, glycolytic, and ATP PCR contributions. So the study hypothesized that the original SIT protocol, which is around 30 seconds of exercise or work, would lead to a greater utilization of glycolytic energy. However, the modified SIT protocols would lead to a greater ATP PCR provision. To determine the ATP PCR system contributions, the fast component of excess post-exercise oxygen consumption, or EPOC, was studied. The fast and slow components of EPOC were analyzed for 5 minutes using bi-exponential and mono-exponential curves to see the best fit for recovery response. The results indicated that the 10-to-4 group had the most optimal work-to-rest ratio to allow for a greater utilization of the ATP PCR system, and that the ATP PCR contribution was greater for the 10-to-4 group as compared to other groups after only training 2 weeks. So ATP PCR contribution precedes improvements in anaerobic performance, and may need a longer training intervention to express enhanced anaerobic characteristics. How does this relate to my chosen field? So, since I want to be a physical therapist, I do believe that the ATP PCR system is extremely important to understand in regard to my patient's injuries. For example, if someone lifted a heavy weight wrong or came in for a basketball, baseball injury, or any other injury for a sport that involves more of the ATP PCR energy system, I will have the knowledge of that energy system that tried to help them succeed in that motion that they were trying to do. I do believe that it is important for any health official to know how these energy systems work, and maybe for more physical therapists to incorporate the different energy systems when it comes to dealing with a patient's injuries, and in order to rehabilitate them, or more exercises involved with the energy systems can be very beneficial when rehabilitating a patient. The following are the sources I use to get my research for this presentation and all my information that I needed. I really did find this system the most interesting, and it's actually one that I use quite frequently, trying to lift weights when I work out, for squats, trying to lift a heavier weight or squat a heavier weight. So I did think this was one of the most interesting ones, since I really don't do any endurance activity, which I should probably start doing more. But yeah, I found this one really interesting. I enjoyed researching about it, and I do think it will be very beneficial as a PT to know how all these systems work. So thank you for listening, and I hope you have a great rest of your day. Thank you.